Good afternoon, everyone, and welcome to today's Lipid Maps seminar. So I'm Ruth Andrew, and I'm your host today, and I'm based in Edinburgh, and I'm really pleased to welcome you to hear our seminar this afternoon. The webinar will be around 45 minutes and will allow 10 minutes at the end for questions. We're very grateful to sponsorship from Cayman Chemicals and for their ongoing support. So I'd now like to introduce our speaker. Today's speaker is Professor Norbert Leitinger from the University of Virginia. He's the Professor of Pharmacology and Associate Director of the Robert M. Byrne Cardiovascular Research Center. His work focuses on regulation of inflammatory and metabolic responses in chronic progressive disease, including atherosclerosis, heart failure, diabetes, and fatty liver disease. He's currently investigating cellular and molecular mechanisms involved with acute and chronic inflammation, and is particularly interested in how tissue damage is translated into chronic inflammation and fibrosis, and how oxidized phospholipids and other danger signals like ATP or heme contribute to these processes. His lab identified the function of oxidized phospholipids as immune modulators, and they first described the redox regulatory macrophage phenotype MOX. He's been involved with developing gene-based therapies and therapeutic approaches to target oxidized phospholipids to prevent chronic inflammatory tissue damage. So overall, Norbert's research is aiming to identify mechanisms that drive pathology in chronic progressive diseases and devise therapeutic strategies to either halt the progression of fibrosis or lead to resolution of the inflammatory response. So we're very much excited to hear Norbert talking today, and I'd like to hand over the webinar to him at this point.
Thank you, Ruth, for the kind introduction, and thank you for inviting me and giving me the opportunity to present our data here. And trying to do today is to give you kind of an overview of what the lab has been up to the last few years and where we go, which directions we're going, and what we're planning to do in the future, focusing on oxidized phosphatidylcholines. And the reason for why we focus on cholines mainly will become evident when I talk to you about the models that we're using. And as Ruth mentioned, we're really interested in how these lipids modulate chronic inflammation and possibly fibrosis. So here's the outline of the talk. I'll talk a little bit, introduce the oxidized lipids to you and the structure-function relationships, but probably not necessary to this audience so much. And then I'll talk about the models that we now use to study the role of these lipids in vivo for the first time. And that's really exciting and pushed our lab towards the next level here with looking at whether these lipids really have something to do with disease progression or not. And then at the end, if there's still time, maybe talk a little bit about how we're going to translate these findings into human pathology. So oxidized phospholipids, at least the ones that we work with, are arachidonic acid containing phospholipids, as you all know, are highly prone to oxidative modification. Here's a mass spec picture of what happens with one molecule, palmitoyl arachidonyl PC. Once it's oxidized, you get a bunch of fragmented oxidized molecules and a bunch of oxygenated molecules. We're able to separate them by solid phase extraction. We'll talk about the long and the short chain fraction of OXB, APC, and we can test them individually. But still, if you look at all the different structures that can derive from oxidative modification of arachidonic acid, it's still a big mess, and there's still a lot of possibilities that can happen and that now are present on the phospholipid. And we found over the years that a lot of these structures have very unique and individual biological functions, and the cells react very specifically to some of these structures with very specific reactions and modulations in cellular physiology. So to make a little bit more sense out of this mess, just the lipids derived from this one molecule, PAPC, we can actually group them based on their structural features. So we have in the full length or the long chain lipids, we have the isoprostane containing lipids, or the hydroxy and hydroperoxy lipids. that would fall into the long chain, and then the short chain, all the fragmented products either contain aldehydes or carboxylic acids or gamma keto hydroxy groups. These were the lipids that were identified by Stan Hayes and to be ligands for the CD36 receptor. And then you can also, if oxidation progresses, you can non-enzymatically produce lyso-PCs as well. And so by grouping these into different groups now, we have a little bit more clarity of what the different functional groups will do on cells. So how do cells recognize these lipids? There's a variety of things. We don't have one specific receptor that we can knock out and eliminate all the actions of these lipids. So it's very heterogeneous of what cells do when they experience these lipids. Of course, these lipids can integrate into membranes and chain membrane organization. They can covalently modify proteins, particularly aldehydes would do that, right? On surface proteins, on enzymes within the cell, and particularly also by modifying KEAP1 induced the NRF2 pathway. There's a bunch of G protein coupled receptors that have been implied to be possible receptors for oxidized phospholipids, the PAF receptor, prostaglandin receptors for isoprostane containing oxidized phospholipids, and some others. And then there is a bunch of pattern recognition receptors that possibly on immune cells recognize these oxidatively modified lipid as danger signals. And among those, as I already mentioned, CD36, Valery Botchkov has shown that LPP and CD14 are receptors for oxidized phosphatidylcholines, thereby interfering with the LPS toll-like receptor 4 pathway. And we and others have shown that the toll-like receptor 2 may be involved in signaling induced by the lipids, but that may also be a function of CD36 being linked to toll-like receptor 2. Anyway, all these different recognition systems cause certain effects in the cells, and one of the major effects which we observed in all different cell types is redox regulation that's modified by these lipids. There's a change in adaptation in cellular metabolism. And in some cells, you get like a modulation of the inflammatory response, sometimes induction of a weak inflammatory response, and a very strong inhibition of a LPS-induced inflammatory based on the interaction with these co-receptors for toll-like receptors. Okay, so oxidized phospholipids, in particular, oxidized phosphatidylcholines, have been implied in a variety of diseases, as depicted here. We are mainly interested in how the immune system is modulated, how that affects cardiometabolic disease. There are indications that these lipids are involved in bone disease, in the vasculature. There's effects of these lipids in the brain and in the heart, and also in the lung, and as I'll show you in a moment, also in the liver. So evidence from for a role of these lipids in human pathology comes from studies by Sam Tsimikas, who has done a bunch of clinical studies showing that the levels of these oxidized phosphatidylcholines correlates with coronary artery disease, and even serves as a predictor for the presence and the progression of atherosclerosis in human populations. So the studies have been done with an antibody based ELISA. The antibody recognizes oxidized phosphatidylcholines, was described by Joe Whitstone first, and gives a nice correlation with disease parameters. However, in all these studies, obviously, we're not able to discern different structural features of these oxidized phospholipids. Other things that this group has shown in humans, for example, that a major carrier for oxidized phosphatidylcholines is Lp(a), which of course we cannot study in the mouse because mice don't have that. So we still don't know what the carriers in mice. So the questions that we're trying to answer in the lab are, how do these oxidized phospholipids, or do these lipids, play an active role in chronic disease, and how do they modulate disease initiation or progression. The reason we're able now to study this in vivo is completely based on the initial finding by Joe Whitstone that this natural IgM antibody EO6 binds to oxidized phosphatidylcholines. It requires the phosphatidylcholine group and an oxidized moiety on the phospholipid, and both together both of these things are required for recognition by this antibody. And so a few years ago, Joe's group showed that, or produced mice that express a fragment of this antibody, SCFV-EO6, constitutively, and he was able to show that these mice were bred on an LDL receptor knockout background, that these mice were actually protected against atherosclerosis and hepatic steatosis and a NASH phenotype later on, so in a separate paper. But this is the initial paper published in Nature, where they show that if the mice overexpress this antibody fragment on the LDL receptor background, in addition to an atherogenic Western diet that causes massive hepatic steatosis and also atherosclerosis, these mice are protected against both in this setting. So that was very exciting and very confirmatory for everything that we had initially hypothesized, that these lipids may play a role in disease progression in vivo as well, right? But there are several questions that still remained open after that study, and that is, for example, again, in that study, they used the EO6-based ELISA to measure the quantity of the oxidized lipids in the plasma of these mice. And so we were still wondering about what oxidized PC species are neutralized and depleted by the EO6 antibody expression, and whether the depletion of OXPC species would be sufficient to prevent the initiation of the disease or the progression of the disease. For example, in cardiometabolic disease or in liver disease, the progression from a steatotic liver into a fibrotic liver. Where do these lipids and which species play a role in the disease process? In order to study this, we would have to induce the expression of this antibody fragment either at the beginning or during the progression of the disease. And as a model to test this, we choose to study fatty liver disease, which has a nice progression from first steatosis to what is called NASH, and then further on to fibrosis. So we thought that by inducing the expression of the EO6 antibody either at the beginning of the disease process or during the progression from NASH to fibrosis, we could study a possible involvement of these lipids in this disease. So the first experiment here we did, so we put the mice on what is called an FBC diet, a high fructose, palmitate, and cholesterol diet, with a lot of sugar in the drinking water. So this causes really massive liver damage over time, progressing to fibrosis at this time point after 20 weeks. So the first experiment what we did, we first of all, we produced a EO6 construct in an AAV8 vector that we could now express or give to the mice, and it also had a Cre dependence on, and if we express that in or injected into albumin Cre mice, we would be able to express it particularly in hepatocytes in these mice. So it's kind of overkill. The AAV8 goes into the liver mainly, and then it's also Cre-dependent expression in hepatocytes. And we did that because we thought this was a liver model. We really wanted to induce it in hepatocytes for this purpose. And then we would give it 2 weeks before we started the diet to let the virus express, and then express the EO6, and then start the diet and go for 6 weeks and see if it has an effect on the formation of hepatic steatosis. And here's some of the results. These are published, so I'm not going to go into too much detail here. You can check the paper if you're really interested. So what we found here is that the virus really expressed the EO6 in the liver. We can detect it. It's MIC tagged, so we can actually detect it by a MIC antibody. But what we didn't know and didn't expect so much was that it was also secreted into the plasma. So in addition to having a liver-specific effect here, we also ended up having a systemic effect. And so at the end of the day, we were not able to discern whether the effects we saw were purely due to the expression in the liver or systemic effects. But that would come in handy later on for the second study that I'm going to tell you about. So what we found is that when we expressed EO6, and that was similar to Joe Whitstone's findings, that we could inhibit and partially prevent the accumulation of lipids in the liver, and the triglycerides would be lower, and also ALT, AST, liver functional parameters for liver pathology were drastically decreased. So protective effect by expression of these antibodies. The expression levels, as you can see from the plots here, varied among the mice, but it was really interesting to see that when we correlated the expression levels in the liver with the triglycerides, for example, there was a really nice correlation with, the higher EO6 was expressed, the more protection there was. So the second experiment we did was, here we wanted to see whether the EO6 or a depletion of oxidized phospholipids during the progression of the disease into fibrosis has an effect, right? So what we did here is we fed the mice for 6 weeks with this FBC diet, which caused massive steatosis, but no signs of fibrosis and very, very slight inflammatory upregulation at this point. And then we injected the AAV8 virus to express the EO6. And then we let it go and fed the mice for an additional 14 weeks to get to the point of liver damage and fibrosis, and see whether the expression of the EO6 during that time would have an effect. And here's what we found, that again, a liver expressed the construct and also secreted into the plasma. And despite the fact that AST and ALT levels were already high at the point when we injected, the further progression of liver damage was inhibited by injecting that virus and expressing the EO6. We didn't get an effect on the hepatic triglycerides because they were already accumulated at that time when we injected. But what we found is during the further progression, there was a striking difference in the size of the lipid droplets, with much smaller and more lipid droplets in the mice that received the EO6. And then looking at the fibrosis, we found that there was less fibrosis in the mice that received the EO6. And when we did gene expression analysis, we found that genes related to inflammation and fibrosis were drastically reduced based in those mice that received the EO6 during the progression. So, which led us to the conclusion that lowering these oxidized lipids would also have an effect on the progression of the disease into fibrosis. So how do we know the expression of EO6 in the liver has an effect on plasma oxidized phospholipids? So to do that, and to also look at which species are actually affected by the EO6, we developed or refined our already existing mass spec analysis method. So what we did here is we used LPP Tiger for in silico oxidation and identification of possible lipid structures that could be formed. We validated that with the lipids that we could produce in vitro by in vitro oxidation of PAPC. And then what we did at the end, we validated what those structures that we could find in vivo in the plasma and in the liver extracts of these mice. I'm going to show you the data from the plasma, and we focused only on molecules derived from PAPC and from PLPC. And here the predicted oxidation products, and then after carefully removing isobaric species and all these other things, we came up with a refined group of here 48 and 35 molecules that we would consistently monitor in the plasma of the mice, either receiving an adenovirus that expresses GFB or an adenovirus that expresses the EO6. And this is what we found in the first experiment. So by testing all the lipids that I just showed you, comparing the GFB expressing mice to the EO6 expressing mice, there was no effect on non-oxidized lipids, on phospholipids and lysolipids, for example. But if you look at the truncated and the full-length oxidized PC species, they're all reduced in the mice expressing EO6, telling us that the antibody had something to do with this. So now the questions, of course, that come up from these analyses is, how does the EO6 do this? How many lipid molecules are bound to the EO6 protein? How does the protein or the peptide eliminate these lipids? These are all questions that we're really interested in. We want to know, and we're trying to study. And another question that, of course, comes up: where are these lipids coming from and how they are carried in the plasma? These are the results from the second experiment where we did the intervention. And again, there was not much of an effect on non-oxidized lipids, but again, a lot of the truncated species and the full-length species were, if anything, decreased in the mice expressing EO6. So what might these lipids be doing in the liver or to the liver cells to promote or to prevent the promotion of hepatic steatosis and other things? So we did gene expression studies and in hepatocytes, and found that the hepatocytes readily recognize OXPAPC species by regulating a total of 1,367 genes up and down. And a lot of the gene regulation was actually mediated by lipid species that were present in the truncated fraction of OXPAPC. And when we did pathway analysis and compare what pathways may be induced by the whole OXPAPC mixture versus the truncated versus the full length, so we found that cells respond to all different structures of oxidized phosphatidylcholines with upregulation of the NRF2 pathway. And we've seen that in all other cell types as well. So this is one of the common underlying responses that cells have to oxidized lipids, but then there's also a lot of other pathways that might be relevant for the pathology that we are seeing. So for example, in the context of NASH and the progression to fibrosis, we saw that OXPAPC and the truncated fraction regulated the cholesterol synthesis pathway, which is relevant for the development of NASH. So to summarize this part of the study, what we've shown is that we can actually, by depleting oxidized PC species in the plasma or the liver directly, we don't know that yet, we can make that conclusion, has a potent effect on the development of hepatic steatosis. And if we try to intervene with the progression of steatosis into fibrosis by injecting the virus and expressing the EO6 at later stages, we can even impact the progression into fibrosis in this disease model. So to summarize these preliminary studies that are ongoing right now, I told you there's a second cohort of mice that's being analyzed right now. And the preliminary data show that cardiomyocytes are responsive to oxidized phosphatidylcholines. They change the metabolism, and some of the inflammatory genes are different also. In the in vivo studies with the albumin-free mice and the EO6 expression on the high-fat, high-sucrose diet showed that the effects on cardiac function, effects on the gene expression in the heart and gene expression in the kidney, and also in the liver, which I didn't show you here. But so there seems to be systemic effects of oxidized phospholipid lowering in a model of cardiometabolic disease in mice. And we also found these very striking sex-specific effects, differences between males and females in response to oxidized phospholipid. Let me just tell you how we're trying to translate these findings into human pathology a little bit more. So what we started doing is using the assay that we developed for mice quantifying oxidized phosphatidylcholine species, adapting that to human plasma. And what we did, our first study was to collect plasma from ICU patients, intensive care unit patients, which actually represent a mixed bag of all kinds of severe pathologies. And we wanted to go into this very unbiased and see, is there any correlation of any of these species with any disease phenotype or at all? And if so, which ones would come up, right? So far we have about 100 patients that we have analyzed. We have plasma from before the first visit and then after, 3 days after, so we can do timelines and all kinds of things. So the hypothesis being that some of these OXPC species may contribute to the pathology and maybe even to the outcome in these critically ill patients. Here the patient characteristics. Here are the questions which we would ask, and we would do mainly correlations with the levels that we measure in these plasmas. One particular thing we were very careful about when we collect the plasma, that we put them in antioxidants right away. So we collect it into BHD, and then the plasma is going to be stored in the presence of BHD, butylated hydroxytoluene, to prevent further oxidation. And we've done studies now over time, over months, that showed that when we do that and store the samples at -80 in the presence of BHD, that we do not get significant further oxidation and degradation of these samples, which is important because we want to collect these samples over time and then analyze them at more or less at the same time. So here's again the procedure. We included our LPP Tiger in silico approach and in vitro oxidized lipids, and then validated the lipid species, now not in mouse but in human plasma. And we find slightly different panel of lipids there that but mostly overlapping and the same species that we can detect in human plasma. Here's an example of the standard curves that we did with the available standards. Obviously we don't have standards for each individual oxidized phospholipids, but we have standards for almost every group of oxidized phospholipids, aldehydes, lysolipids, beta gamma hydroxylipids, and everything. So the standard curves look very similar, which tells us that even if we measure something that is, maybe not ionizing the same way or has slightly different behaviors in the mass spec, at least it falls into that range, and we can get a pretty good estimate of changes in the quantity of that particular lipid species. So here's some of the species that we actually analyzed in the human plasma with the different features and their concentrations. We're usually in the low to higher micromolar range, each of those individual species. So what we can do then is we can do a hierarchical cluster analysis of the different lipids that we measure. And it was very nice to see that there were actually 4 main clusters in the plasma of these patients that came out. One was more like a carboxylic acid containing cluster. Number 3 was like an aldehyde cluster, keto groups, and the lysolipids would cluster together as expected. And then here's also an aldehyde cluster, mainly short chain. This one is mainly long chain. This is mainly short chain here. So by just taking now unbiased these 4 clusters and correlating each cluster with disease and pathological parameters in these patients, we get something that looks like this. So we have the different clusters here, group 1, 2, 3, 4 on the bottom. We also can manually just select all the truncated lipids, all the full-length lipids, correlated all the aldehydes that we defined, all the carboxylic acid containing, all the gamma ketro and lysolipids and the non-oxidized. So what was immediately evident when we looked at the beginning about like 50, 60 of these patients, there was a very strong correlation with two disease phenotypes, one being kidney here and then one being liver. So now this was very interesting to us and very confirmatory that some of these lipids may have something to do with these pathologies. The reason other pathologies would not show up yet, I mean, we hypothesized there might be something with lung disease as well, and metabolic syndrome and obesity, is because some of the groups don't have a high enough N to show anything. So there's some patients, but probably not enough. Also with sepsis, for example, we didn't have enough sepsis patients in these to get something significant here. Nevertheless, so this is very exciting for us, and we're going to expand these studies into other patient populations and continue collecting plasma from ICU patients as well. And so in conclusion, I can say what I showed you today, that these OXPCs are present in the plasma of critically ill patients, and the concentrations are in the micromolar range for some of these. Within the cohort that we analyzed, we doing the cluster analysis that similar modifications correlate with each other. And then some of these species actually, or groups of these species, associate with certain clinical parameters. And we're going to expand that. And hopefully I'll be able to report more on this soon. So that's it, what I wanted to tell you today. So wanted to thank the people who actually did the work, Clint, Scott, Katie, and Anna, our collaborators. Alex provided the plasma from the ICU patients, and a lot of more students and collaborators here at UVA, and NIH for the funding. And I thank you very much for your attention, and I'll be happy to discuss and answer any questions that may have accumulated during the presentation. Thank you.
Thank you very much, Norbert. That was very interesting and very exciting to see some of your new data coming through there. Maybe I could start with the questions. Your sex-specific differences were really striking in your gene expression. I might not have picked it up, but did you find sex-specific differences in the phenotypic responses to the antibody treatment as well, or was it more when you dialed down on the actual the gene expression?
So far, these are preliminary. So far, they just came out by analyzing the gene expression data separately. Mm-hmm. That was the first thing we have. So the phenotypic responses, we will. Oh, you're talking about the heart stuff. And so we're digging into that right now. We want to see if that holds up due to in the function as well.
Yeah, no,
I don't know. I can't tell you yet. Yeah.
No, that's fine. I was thinking that was thinking as I went through. Now we're getting a little flurry of questions here, so maybe I could ask Patricia Prabutsky.
Perfect. Yeah, thank you very much for the very interesting talk. Maybe you can clarify one thing for me, which is you suppose that the OXPC is bound to the antibodies because it's going to be cleared by the antibody. Am I correct in this representation? So it's clearance, basically?
Yeah, I mentioned during the presentation, this is one of the things we really don't know how it happens. So we obviously get lower levels, and the first conclusion would be there's some induced clearance after binding the antibody fragment. Whether this is really happening or not, or is it sequestering it so that we can't detect it anymore, we really would have to show that experimentally. So far, I don't have an answer how it works. And also the stoichiometry doesn't make quite sense by like a 1-and-1 binding. One lipid binds to one antibody fragment and then it's cleared. That would not make sense. So one other hypothesis I have that might be happening is that the antibody fragment binds a whole bunch of lipid at once. How is that possible? Maybe a vesicles that are floating around that contain many of these oxidized lipids, and then the whole vesicles are cleared, whole lipoprotein particles are cleared, which might be happening in the kidney. These are things we're thinking about doing, like labeling studies where we can actually trace it. The other big question is where do these oxidized lipids come from, right? Are they coming from the liver, from other organs? Why are they accumulating in the plasma and stuff like that? So we're thinking about labeling studies and trying to trace it.
Because my initial question was coming from a different angle, which I suppose is also highly hypothesizing. It's more to the function of different other classes. So if you could quickly tell if you have any idea if you think that the oxidized phosphatidylcholines have the same effect like other lipid classes might have, or do you have any plans for future investigations? Sorry, and thank you.
Okay, so other classes you mean by phosphatidylserines and ethanolamines? They have been shown they have biological activity for sure, and we have done some studies to compare them, and there are other people who have done studies with ethanolamines, and yes, they're also biologically active. The only reason I'm focusing on PCs right here is because the antibody only requires the PC group. It wouldn't affect all the other phospholipid classes. So that's our model, and that's why I'm focusing on that. But you're absolutely right. The other oxidized phospholipid molecules, phosphatidylserines, will have effects on cells. Will recognize those moieties as well.
Thank you very much.
Okay, thank you. So the next question is from Elena Ambrogini.
So I am interested in understanding the role of oxidized phospholipids in insulin resistance, because I work with the mice provided by Dr. Wisdom, and this is something that I also see. And so E06 SCFV transgenic mice have increased insulin sensitivity during aging, and they respond. They don't get so much insulin resistance when they age, even when fed a normal diet. And what do you think it could be a possible link?
It's a difficult question. So first of all, we didn't see much on insulin resistance when we fed the mice the FPC diet. This seems to be really strong on liver, and so we didn't see much of an effect there. That's why we neglected it for a while. Now with the high-fat, high-sucrose diet, we see an effect, as I showed you, right?
Yes.
So now the question becomes, we're looking at liver, which may be part of it, so protective effects on the liver to begin with. But if you talk about insulin resistance, one possible place where that may play a role is obviously the adipose tissue, right? So we haven't looked at the adipose tissue yet, but I would assume that since all the other organs we looked at are affected, maybe there's a protective effect on the adipose tissue as well.
Yeah, because when I age the mice up to 22, 24 months, obviously I study bone, right? So I'm interested in the aging.
Yeah.
The mice really gain less weight.
Yeah.
That's a clear thing. And I did a glucose tolerance test around the age of 16 months. And they handle the glucose much better than wild type mice. And so I think I'm just interested in understanding better this phenomenon, and I was wondering if you have any particular target that I should look at it.
So the thing with if you have differences in weight gain already, it's very difficult to discern what affects the glucose tolerance, right?
You don't see a difference in weight, but you see a difference. Your weight, I think, looks the same, but you still see much more decline in glucose when you give insulin, right?
So maybe this would be the model to look at where it's occurring, right? With the same weight. Yeah, maybe we should look at the adipose tissue. Maybe we can collaborate.
I think there is something. Again, my expertise is on bone. I'm an endocrinologist, and so I should be able to understand this. But I think it's something that is worth to look at it.
Absolutely.
Because it's a very striking effect, I think.
Cool. Thank you.
Thank you, Elena. Okay, our next question is about human translation from Tong Shen. Tong, do you want to join?
Yes, hello. I'm asking, seemingly like the lowered oxidized PC is kind of like the byproduct or result of the antibody EO6, which will be more promising, like to translate to human. Shall we introduce EOC 6 or try to lower oxidized PC like via antioxidant treatment, which would be more like progressing better?
So antioxidant treatments didn't work, right? People have tried that before. So that's for us working with these lipids, that's really exciting that we have now a tool with the EO6 to lower lipids. How translatable is that into humans? I know that Joe Whitstone has a lot of ideas of how he wants to develop treatments based on that. Whether the AAV, the gene transfer thing, would be something that is translatable. You know, there's a lot of problems with AAV. I don't know. But it's definitely a good tool to specifically target these. It seems to work very highly specifically for OXPC. And the other thing is also, I really would want to know first how, going back to the first questions here, how it actually eliminates the lipids and what the pathways are and what the exact mechanisms are. And maybe that would give us some more of a clue of how to translate that into a human system.
And another question, if I may, will be like in the beginning, the introduction saying it is specifically bound to ApoB or lipoprotein A. So in the liver, oh, the mice doesn't have it. That was why it wasn't checked.
In humans, it's carried. So Sam Chimika showed that in humans, it's carried on Lp(a).
Only. I see.
Not only, but the majority.
A majority.
Majority, right? It's also present.
Is PAPC the most abundant oxy-PCs? Like other, like choosing that as representative of oxidized PC is by abundance?
I think PLPC would be the most abundant. I think linoleic acid is a little bit more abundant in the plasma. So that's why we included it also in our mouse study. But there are fewer oxidation products. I mean, since we know a lot about the PAPC oxidation products already, we also included that. And I don't think it's a matter of where it comes from. It's the sum of all the functional groups that come from either linoleic or arachidonic acid or other polyunsaturated fatty acid oxidation products that cause the effects, right? So it's all the aldehydes, for example, all the short chain aldehydes coming from whatever precursor lipid that would cause the effect. Which also means all the measurements that we are doing, we are massively underestimating the total amount of these lipid oxidation products that are floating around there.
Yeah, it's representative. Cool. Thank you so much.
Thank you, Tom. Our next question is from Ellie Bietianai. Hello.
Thank you for sharing these interesting results.
Yeah.
I wonder, have you considered the role of exosomes in the delivering of this oxidized lipid? Recent data coming from different labs have shown the presence of this oxidized lipid and other oxidized end products within exosomes, so they can contribute to the spreading of the oxidized stress, but they can also spread the protection mechanism that might be involved in the maybe liver and cardiomyocytes you have presented. Thank you.
Absolutely, yeah, that's a great point. And I mentioned that one of the possible mechanisms that how it works, that a bunch of lipids can be eliminated by one peptide, could be because they bind to, I call them vesicles or whatever membrane blebs or things that are pinched off from any kind of membrane would contain these oxidized lipids, and exosomes. I don't know if the lipids would be carried inside the exosomes or they just would be parts of the membrane of these exosomes. Either way, it's an interesting thought that this is one of the major transport mechanisms in the plasma.
Thank you. Thanks, Lily. Our next question, we might have touched on this before, but Stella, would you like to ask your question?
Thanks, Norbert, for the presentation. We've previously worked with some oxidized lipid panels, and what we found, and which agrees with what you say, is that we have different panels in different diseases. So for Alzheimer's disease subjects, we found different panels, groups within the oxidized phosphatidylcholines, and for chronic kidney disease, different panels, and for periodontitis, different panels, and in the brain of mice that were fed with high-fat diets, different panels. So my question is this EO6 generic and targeting the Ed group of phosphatidylcholine, or is it targeting maybe the aldehyde group or the long chain, the truncated ones? Because what we want to progress to is to check what would be used to target the ones that are upregulated or downregulated in specific diseases. But the presentation you've given is showing us if you are using this EO6 to target all the oxidized phosphatidylcholine groups and not just specific groups. So how specific would this be, or will it just be like one size fits all the phosphatidylcholines?
So this is the question that we had going into this study, right? So we wanted to know which species are particularly recognized. So Joe Whitstone has shown, when he described the antibody, that what is required for the antibody to bind is the PC group, the PC head group, and an oxidized moiety. And that could be either a fragmented aldehyde containing or something group that is recognized at the same time with the PC group, or a long chain lipid. And the explanation for that is usually the long chain oxidation groups, they're folded back like in an isoprostane kind of fashion. So you would end up having kind of a short chain epitope, if you will, together with the PC that's now recognized. So I don't know if you can say it will bind all of them, but the panels that I showed you, like in 4 different experiments, we have certain species that are really strongly eliminated and others that are recognized less well. We couldn't find a pattern yet. I mean, definitely long chain and short chain, both are eliminated, but we couldn't find a specific pattern to say that, oh, okay, only aldehyde containing ones are preferentially cleared or something.
Yeah.
Doesn't seem to be a specific pattern.
Okay,
yet.
Thank you.
Thank
Thank you.
We've got time for just one last question from Samar. Okay, I can ask it. So Samar's interested in the fact that PCs are also parts of sphingomyelins and wondering whether the PC that's oxidized that comes from the sphingomyelin is included in the measurements you're making in plasma.
It's a great point, and I think we have done full lipidomics as well, and we have the sphingomyelins in there, and we didn't see a difference in the sphingomyelins.
Okay.
So when we do the targeted analysis that I showed you, we only have phosphatidylcholines in there, but in the full lipidomics, and that's part of the first paper also, we have included all these other lipid species.
That's fine, Martin. Check that there. Well, I think it's just to finish up to thank Norbert very much for an absolutely fantastic seminar and sharing both his published and his new studies, which we look forward to seeing coming through soon. So goodbye. Thank you.
Thank you.